CRISPR is a revolutionary technology that has the ability to edit genomes in a precise and inexpensive way. It has been hailed as a game-changer in the fields of medicine and agriculture, with the potential to cure genetic diseases, create disease-resistant crops, and prolong the shelf life of perishable foods. However, with the advent of CRISPR also come ethical concerns. One of the most notable examples of the ethical concerns surrounding CRISPR is the CRISPR babies experiment conducted by Chinese researcher He Jiankui in 2018. He used CRISPR to edit the genomes of IVF embryos, resulting in the birth of 3 babies with altered genomes. The goal of the experiment was to make the babies resistant to a potential HIV/AIDS infection by disabling a gene called CCR5. The experiment sparked a global debate on the ethics of genome editing in embryos. Some criticized Jiankui for being reckless and trying to play God, while others argued that the experiment was a necessary step in the advancement of CRISPR technology. The experiment also raised questions about the potential long-term consequences of genome editing, and whether or not it is appropriate to create designer babies with superior genes. Another concern is the potential for CRISPR to be used for eugenics, or the practice of improving the human race through selective breeding. If gene editing becomes more widespread, it could lead to a divide in society between those with superior and those with inferior genes. The idea of eugenics is not new, and the dangers of using science to improve the human race have been demonstrated throughout history. In the early 20th century, several countries, including Canada, Denmark, Switzerland, and the United States, passed laws that allowed for the compulsory sterilization of those deemed unfit to reproduce. The most infamous example of eugenics in action was the Nazis' Action T4 program, in which doctors administered mercy killings to those deemed incurably sick, resulting in the deaths of between 275,000 and 300,000 people. In the United States alone, it's thought that around 60,000 people were sterilized based on the misguided theories of eugenics. Sadly, a large number of those deemed unfit were minorities and immigrants. However, the impact of eugenics went much further than many realize. Poverty proved to be a significant factor in who was deemed unfit, regardless of one's color. Thousands who would probably simply be classified as having a learning disorder today fell into this category. In some parts of the country, people with illnesses such as epilepsy were effectively banned from marrying and procreating. Eugenics is a dark chapter in history, one that serves as a reminder of the dangers of using science to improve the human race. The atrocities committed under eugenics serve as a stark reminder of the dangers of allowing the government to dictate who should and should not be allowed to have children. As we continue to develop new technologies such as CRISPR, it is crucial that we learn from the past and ensure that these technologies are used responsibly and ethically, because CRISPR has the potential to bring about significant benefits in medicine and agriculture. In medicine, it can be used to cure or ameliorate the symptoms of a wide range of genetic diseases, including blood disorders, cancer, and ocular disorders. In agriculture, it can be used to create disease-resistant crops, prolong the shelf life of perishable foods, and increase their nutrition values. Currently, genome editing with CRISPR is only accepted in somatic cells to ensure that any potential consequences cannot be inherited. However, as the CRISPR babies experiment demonstrated, it might be possible to edit the genomes of children in the future. Therefore, it is crucial that the ethical concerns surrounding CRISPR are addressed, and that appropriate regulations are put in place to ensure that the technology is used responsibly.